This conference will now be recorded.
So before we start, I just want to confirm like if everyone can hear me. Am I audible to everyone? Once again, I just want to confirm if everyone can hear me. So yeah. So a very good morning or evening to one and all, to all the speakers, supporting associations, students, and delegates present virtually with us today. Myself, Jessica Reeves, Conference Secretariat of Coalesce Research Group, very delighted to welcome you all. Even during this pandemic, it gives me immense pleasure to having meet you all virtually at this webinar organized by Coalesce Research Group. So, giving a brief introduction about ourselves. So, yeah, we are from Coalesce Research Group, which is an USA based emerging open access publisher and international conference organizer, which ove to serve the scientific community by serving the high quality, genuine research work from eminent scientists, professors, scientific research communities in all fields of medicine, clinical, life science, engineering, and technology to the research scholars, students, libraries, educational institutions, research centers, and industries with an aim of enhance your research in the respective fields. Recently, we have also started our own platform for open access peer reviewed scholarly literature where all the content is freely available without any charge to the reader. We aim to share the new research advances through scientific publications, which will help in minimizing the borders and contributing the use of research works of eminent authors, scientists, researchers, research scholars, students, etc., across a broad spectrum of medical, clinical, pharma, and engineering scientific disciplines and its subspecialties to promote the ideas which will guide the development and facilitate the use of scientific information and ensure its quality and also will be connecting link between the researchers and scientific community. We are committed in ensuring that our journals are disseminated worldwide so that the researchers are able to access our content wherever and whenever they need it. We would be very glad if any of our speakers or delegates participating in today's webinar will be interested in publishing any of your research papers in an inaugural issue in any of your interested journal. We'll also send you an email with all the details of the journals after the commencement of the webinar. Finally, we thank each and every one of you, the participants at this conference or the webinar, we hope you enjoy the conference and your participation will definitely contribute in new collaborations and research initiatives. So now, starting with our today's session, uh, so checking online, we do have uh, just a moment. At present, we actually have our poster presenters, Dr. Francisco Capre from Plant Molecular and Cell Biology Program, USA. And I just want to know, like, if you will be able to present your poster.
Hey, can you hear me?
Yeah, we can perfectly hear you.
Um, I would be able to present in a couple of hours, ideally.
Okay.
I do have some pre-existing
Hmm.
commitments for this morning.
Okay, I understand. I completely understand.
Yeah, sorry about that.
No
But
problem.
I
That's
can I can
completely
rework
normal.
my schedule to anticipate a little. I just cannot do it right now, though.
I completely got it. It's okay. I got it. So yeah. Now, do we have any oral presenters online so that we can have your schedule? We are really sorry for the technical issue. Just because of the technical issue, like our schedule has got upside down, and yeah, some of our speakers are missing right away. So if any of our speakers are online, yeah, you can take an initiative to start your session right now.
Hi, Elena.
Yeah,
Hi, I'm
yeah,
Monica Matias.
yeah, Dr.
I,
Monica Matias. Yeah,
I am presenting a poster,
yeah,
and I can
sure,
do right now if you want.
sure. Thanks a lot. So yeah, we have made you the presenter, so you can just click on the screen button so that you can display your poster with us.
I have a yes. I did a presentation of my poster. Is okay?
Yeah, yeah,
Yeah. Okay.
yeah.
Ay, what? I don't know if it is
Yeah,
okay. My
it's visible. It's visible. It's completely visible.
yes. I am trying to move the.
Okay, you'll have three dots over there on the screen. You can just move that to move your thing. So
Ah,
that is
okay.
actually not visible to us, so it will be only visible to you.
Ah, okay.
Yeah,
Okay. I can. I have 50 minutes.
yeah,
15 minutes.
15. Yeah,
Okay. Well, the title of my work is a SNP GBS single trait genome-wide association study in a collection of Avena sativa genotypes. I don't know how to pass the.
you can just press the arrow buttons so the slide will move on. Like on the keyboard, you can just click on the arrow buttons.
I see. This is my poster, and I divided in different slides.
Yeah,
We conducted a single trait genome-wide association study in oat, Avena sativa genotypes. The collection is a diverse origin and constitute a representative sample of the germplasm most commonly used by the INEA Chile oat food genetic breeding program. Our hypothesis was that variability in the studied germplasms would be large enough for detecting useful associations among SNP markers and phenotypic traits. The objective of the work was to study the associations among SNP, GBS marker, and phenotypic traits in a collection of 190 oat genotypes. So it's not. I see it's working. This is the geographic origin of the germplasm. The 35% is from Chile and 44% from USA. is a collection constituted by cultivars, advanced lines, all F generation F7 pure lines. The method, we establish field trials at 4 locations in southern Chile in 3 years using an augmented design with 3 replicated controls. The traits were data of grain yield, heading time, plant age, loading, and grain milling quality traits that were collected during all the cycle of the plant. We use SNPs markers that were generated by Illumina next generation sequencing platform at Cornell University. A total of 40,000 SNPs was detected, and only 8,000 were used in the GWAS. The methods, the trait data were analyzed with a Spremel software using a mixed model approach with spatial correction. We used 2 models. We obtained adjusted means with the model 1, using the genotype as a fixed effect, and a model 2 that we used to obtain BLAP predictions and heritability, narrow sense heritability, where we use the genotype as a random factor, and we include a correction for the pedigree. We use the StatGen GWAS R package to run the associations among markers and phenotypes. The sequence reads of significant SNP were aligned with the reference genome of Avena sativa released by PepsiCo. This is a table with the OA trait that we measure with the units, grain yield, hectoliter weight, and several traits of milling quality, loading of the plant, and stained grains in different severities, protein and beta-glucan. Well, the main result is that the models, the statistical model used converged in the majority of combinations of location year, being better than the non-spatial model. The narrow sense heritability was moderate to high in the majority of locations and traits, indicating a good quality of the data. 139 SNPs were significant. However, only 16 were selected based on their repeatability in different environments, magnitude of the effect, heritability, and complete matching with Avena sativa PepsiCo. Several undesirable associations were observed considering the objectives of the breeding program. This is a big table, but I want to show you some details. We selected the moderate to high heritability values that this data is here. These are the effects of the markers. This is as the SNP marker and the locations and the traits. We observe that some markers have positive effects, like a high hectoliter weight, and others have undesirable associations. For example, high hectoliter weight but a low yield. Several SNPs were associated to negative effects that are in red letters, and the positive effects are in blue color. The SNPs exhibit significant association with phenotypic traits. However, they were specific to particular environments. The blast of the majority of SNPs to the oat PepsiCo genome resulted in a complete matching, being the SNP preliminarily assigned to 10 different chromosomes. The upstream and downstream sequences of the SNPs selected are being analyzed to assign them to known sequences and primer design. The conclusion is that the variability of the germplasm was enough to detect significant association of SNPs with hidden time, hectoliter weight, plant height, lodging, grain yield, and stained grain, but not with other traits that are very important for breeding program, like beta-glucan or protein, or, for example, the hullet grain extraction. And these are the acknowledgements. This work was funded by Corfo by an Innova project. And I want to say thanks to the Quaker International Oat Nursery by the germoplants provided. And this is my presentation.
Thanks a lot.
I hope that
Thanks a lot for your presentation.
Okay?
Yeah, so, yeah, please continue.
Yeah, are we open for questions?
Yeah, I was actually about to do that. So do we have any questions? Yeah, please, you can start with
Yeah,
that.
so I had a question. In your second to last slide, you said that you're designing primers on genes. To what end? For what purpose are you designing those primers?
The idea of this work is to develop markers for using in the breeding program. I am breeder in the Chilean breeding program of oats, and is the objective to study the oats, but at the same time to generate markers for positive effects. And years ago, the PepsiCo genome reference genome was not available, and now we have availability of the sequences, and we can align the sequence reads and to search in upstream and downstream DNA of the oat, and to develop primers, and then to check the primers in progenies and to validate the results, because this work was made in a collection of diverse origin oats.
That's very nice. So how many markers do you think would be a good subset for you to do this kind of work? Because I know when I was working in Pear, we were using about 10 markers to screen for a specific disease. We lowered it to 3 total markers. How many markers are you aiming for? What would be considered a good result in this specific crop? Because I'm not very familiar with it.
I don't have a number, but I think the ideal is to have a higher number of markers. In this work, I think we have 139 SNPs that were significant, but I chose 16 because these SNPs were repeated in different environments. I think minimum 5 markers for traits, I think, to develop primers, or more, but in this case we have a little number. From 50,000 SNPs, we only select 16. I think maybe it would be very, very good to have more markers, but in this case, I think the characteristics of the trials, we work it with augmented design, and there is noise in the data. So if we work with a higher size population with a better field trial design, we could obtain more markers. But in this case, we have only 16, and only 4 of them have exhibit association with positive effects for breeding purposes of oats in Chile.
Nice, thank you so much. That was a very good answer. Thank you.
Do we have any other questions? So yeah, I guess there are no questions, Dr. Monica, and yeah, thank you for your presentation.
Thank you very much for hearing my presentation.
Thanks a lot. So yeah, moving on to our presentation, we have Dr. Giuseppe Antonio Malfa from the University of Catania, Italy. Yeah, the screen is perfectly visible.
Hi, can you hear me now?
Yeah, we can hear you.
Okay. Say hello to everyone. And last moment. Do you see my presentation? I think you see GoToMeeting too, no?
No, that'll actually
Okay.
not be visible to us, so it'll
Okay,
be
so
only
you can
visible to
see
you.
only the presentation. Okay, good morning.
Yeah.
So I will talk about the antioxidant properties and the biological activities of extract from Betula etnensis. Okay, the genus Betula belongs to the family Betulaceae and consists of about 6-5 species. It is widely distributed in all temperate regions of the northern hemisphere, but it can occasionally be found in the southern hemisphere, especially in South America. Plants belonging to this genus vary in size from small shrubs to some of the largest deciduous forest trees. Distinct species can be found in North America, in Europe, and Asia. And some of these appear to fill similar ecological niches on separate continents and have many superficially similar characteristics. Beeches are among the most attractive trees known for their autumn colors and contribute to the amazing foliage in eastern North America. Many species are adapted to extreme soil and climatic conditions. For example, they prove to have an exceptionally wide side amplitude. They can be found on poor sandy or logy soils at altitudes of 2,000 meters above the sea level in the Alps, for example, like Betula pubescens, and at 4,000 meters of altitude in the Himalayas, like Betula utilis. They even provide the most frost-resistant woody species. Beech trees are fast-growing and therefore important pioneer tree species for afforestation. Etna birch, in particular, is an arboreal essence whose morphological characteristics suggest a source from Betula pendula root, a very similar species present in different regions of Italy and Europe. The geological evidence suggested that southern Italy and Sicily represent an area of refuge for the beach during the last glaciation between 70,000 and 10,000 years ago. And later, in the post-glacial period, they allowed the re-expansion of its range. No longer finding the right weather condition, in Sicily it was confined only on Etna, the volcano, with its altitude about 3,300 meters above the sea level, as a favorable climatic zone. The genetic isolation from the population present in the rest of Italy and Europe and the strong selective pressure due to the volcanic nature of the territory and the Mediterranean climate have made it possible to differentiate in a new endemic taxon, precisely Betula etnensis ruffin. It is, in particular, is an arboreal species, like the others, that reaches heights from 4 to 15 meters, but at altitudes above 2,000 meters often assume a scrub bearing. It's recognized by a white smooth bark, rhomboidal and subtriangular shaped leaves, less sharp than those of Betula pendula with a crenate margin. The appearance of the species is unmistakable due to its tremulous foliage and the white bark with brownish black spots that flanking and curling in a horizontal cylinder persistent in the trunk. The flowers are grouped in inflorescence with the yellow colors, the masculine ones, and the light green, the feminine ones. The anthesis is in the period of April-May, and the fruits are small conical anthers of brown color. Today, due to this characteristic, Betula etnensis is considered a pioneer species because it is able to colonize incoherent lavas, forming woods of a certain consistency, both in pure cultivation and in association also with beech. An area of particular interest for the observation of these amazing tree species is around the Sartorius Mounds, a complex of baton craters in the northeastern side of the volcano. Man has utilized beaches for diverse reasons that include folklore and witchcraft, drinks, essential oils, or simply as a firewood. However, the greatest use are for construction and ornament. The bark of many beeches is highly water repellent and resistant to degradation. Thus, it can be used for covering for boat hulls such as canoes. The healing properties of Betula bark have been known for a long time in traditional medicine in different areas of the world. Several species of Betula have traditionally been used for the treatment of various inflammatory diseases. Crude extracts, fractions, and phytochemical constituents show a wide spectrum of in vitro and in vivo pharmacological activities like anti-inflammatory, antimicrobial, antiviral, antioxidant, and many others. Moreover, today there are convincing evidence in experimental animal models in support of their anti-tumor effects. However, it would be valuable to further investigate the biological mechanism of the different biological active compounds. Several species belonging to this genus are widely used in traditional medicine, including Betula etnensis in Sicily. Previous study conducted by our group have indicated that Betula etnensis polyphenolic extract showed a strong bioactivity against, for example, Staphylococcus pneumoniae, and furthermore, extract from bark contain considerable amount of polyphenols with a considerable radical scavenging activity, and also exhibit a significant ability to different modulate the oxidative stress in an in vitro model of colon cancer, inducing a type of necrotic cell death called ferroptosis. This article was published last year in the International Journal of Molecular Sciences and was presented at the 24th World Congress on Advanced Oncologies. But The purpose of the study I will show you in this occasion was to perform a preliminary phytochemical characterization of an alcoholic extract in order to evaluate some in vivo effects in an experimental model of diabetes induced in rats by the administration of streptozotocin. To this end, together with general parameters such as blood and glucose and insulin levels, amounts of water, etc., we assessed some oxidative stress biochemical markers of some organs involved in oxidative stress, induced diabetes, and in associated complications. For the extract, the total phenolic flavonoid content assay showed values of about 57 milligrams of gallic acid equivalent for gram of extract and about 6 milligrams of catechin equivalent for gram of extract. And furthermore, betulinic acid was also identified and quantified by HPLC-DAD analysis, and it was quantified with a value of 0.069 milligrams per milliliter. The chromatogram for HPLC for betulinic acid, this is our extract and this is the reference standard. Diabetes streptozotocin induced displays all the hallmarks of functional and biochemical alteration observed in diabetes in humans, such as increase in food and water intake and body weight loss due to the degradation of substructural proteins. The present study showed that the administration of bark extract for 28 days significantly reduced food and water intake, as you can see in the table displayed in the presentation, and as well as polyuria, but above all improved body weight loss, a mark of a more efficient metabolic homeostasis. Moreover, we know that in diabetes, the moderate generation of a reactive oxygen species, ROS, mainly due to hypoglycemia, leads to oxidative stress in organs and tissue. The unphysiological high levels of ROS and of reactive nitrogen species, RNS, and the concomitant decrease in endogenous antioxidants generate cellular and tissue damage, so promoting the development of diabetes-associated complications. Induction of diabetes in rats with the streptozotocin resulted in a significant change in oxidative stress markers, both in plasma and tissue. In fact, in our study, it was observed that the administration of streptozotocin induced a massive reduction in non-proteic free thiols group amount in plasma, as you can see here. Liver and both at pancreatic and renal level, you can see also here. The main constituent of non-proteic free thiol group, RSH, is glutathione. Glutathione, GSH, is the main antioxidant. The extract has been able to counteract the depletion of this important endogenous antioxidant. In fact, in diabetic animals treated with the betula extract, the pancreatic, hepatic, and renal levels of GSH were comparable to the values found in control rats. Also, the observed increase in lipid peroxidase in diabetic rats was significantly, counteracted by the oral treatment with extract, together with the enzymatic activity and expression. This is the activity and this is the expression of gamma-glutamyl cysteine synthase, the enzyme involved in glutathione synthesis. But ROS are not the only responsible for oxidative stress. I said before, RNS play an important role in supporting this harmful condition. The result of the present study demonstrated that nitrate and nitrate plasma level directly correlated with nitric oxide production were strongly increased, confirming its overproduction after following hyperglycemia injury in diabetic untreated animals. Differently, in Betula ethnensis treated diabetic rats, the daily administration of the extract induced a significant reduction of nitric oxide levels in all the analyzed tissue, but in particular at renal level. Under the same experimental condition, the expression of two of the enzymatic proteins responsible for the synthesis of nitric oxide has also been evaluated. The endothelial nitric oxide synthase and the inducible isoform, both involved in diabetes-associated complication. The result in untreated diabetic rats showed that the endothelial isoform is strongly reduced, while the inducible isoform displayed a significant increase in protein expression. Additionally, the extract was found able to counteract these reverse effects on endothelial and inducible NOS isoform compared to the controls. Furthermore, the expression pattern of INOS reflects the result concerning nitrate and nitrate levels, suggesting that the marked increase in nitrate and nitrate found in diabetic animals can be linked to the overexpression of this particular isoform. Another important enzyme implicated in elevated oxidative stress condition is M-oxygenase, or commonly called HO1. It is well known that together with its potent antioxidant effect, the increased activity of HO1 is able to counteract the development of diabetes-associated complication. In the present study, HO1 expression was evaluated in pancreas, liver, and kidney. Betula extract, according to previous results, was able to induce this enzyme in all the studied organs, further confirming the protective effect exerted by the extract on oxidative stress diabetes induced. It's clear that the administration of the extract induced the expression of this enzyme. The same thing we found in the other study when the extract was able to induce ferroptosis through the activation and increased expression of HO1. After this, we focus our attention on mammalian sirtuins, a class of protein with important function, including the activation or deactivation of enzymes involved in lipid, protein, and carbohydrate metabolism. It has been reported that oxidative stress may induce sirtuin impairment, particularly SIRT1, so affecting many stages of glucose metabolism in different organs and tissue. Moreover, SIRT1 is a crucial player in the prevention of oxidative damage via variety of mechanisms. Determination of nuclear sirtuins activity in the different organs evidenced that the oral administration of betula extract was able to counteract the streptozotocin-induced decrease in sirtuin activity, restoring level to those observed in the control group. This last result suggests an involvement of SIRT1 activation in improving insulin sensitivity following extraoral administration. Concluding, taken together, the year reported results suggest that the protective anti-diabetic effect of beta-lactam's bark extract are mainly due to the capacity of restoring redox homeostasis through the modulation of some regulatory enzymes that are involved in the antioxidant response, and also through the modulation of sirtuin activity, this important class of protein, which is strictly linked to glucose homeostasis and also on oxidative stress mitigation. One thing before to close, this study has been published few weeks ago in Antioxidant, a journal, an international journal specialized for study related to the science and technology of antioxidant. And at the end, I would like also to thank all the components of the scientific group involved in this study. And also I wish to thank the organizing committee for this scientific event and all for you for your attention. Thank you very much.
Thanks a lot, Dr. Antonio. So, yeah, do we have any questions? So I guess there are no questions.
It's okay. Thank you.
Thanks a lot for your presentation. It was really informative.
Thank you.
So, yeah. Now, next we have Dr. Francisco Capai from Plant Molecular and Cell Biology Program, USA, ready for his presentation.
Ready. So let's see. Wait a second. I need to change the. There we go. All right. Hopefully y'all can see me and hear me pretty well.
Yeah, that's
Excellent.
perfect.
All right. So let me see if I can share my monitor one. Perfect. There we go. You should be able to see my
Yeah,
PPT pretty well.
yeah, that's perfect.
Okay. Actually, I need to share my other screen, I think. Just a second.
Sure.
It's the first time I use this platform. I usually use Zoom. Stop sharing screen. Screen. Share screen of monitor 2. There we go. All right. Now you should be seeing the right screen.
Yeah.
Fantastic. Fantastic. All right. So, well, first of all, thank you so much for organizing this. I organized a few events in the past myself. It's always honestly quite outrageous when the first speaker doesn't show up. So unprofessional of them. And I hope they have a very good reason for not being able to make it in time. But anyhow, having said that, today I will be talking to you about my project. So let's get rolling with it, which is unraveling the genetic architecture of firmness and other machine harvest traits in autotetraploid blueberry using a QTL analysis approach. So thank you for introducing me. I am Francisco Capai from the Plant Molecular and Cellular Biology program at the University of Florida in the US. So today you'll hear a lot about blueberry. The scientific name of this species is Vaccinium corymbosum, which is a deciduous shrub in the Ericaceae family. Now, the true species is northern highbush blueberry, which originates in the northern United States, but is not the one we work with at the University of Florida. What we work with is called southern highbush blueberry. This subdivision was created in the late 60s, crossing northern highbush blueberry with a few southern adapted Vaccinium species. So the bottom line is southern highbush blueberry has a very diverse genetic makeup. However, it is tetraploid, just like northern highbush blueberry, which makes it a little bit challenging in terms of genetics and mapping. But why do we care about blueberry at all? Well, blueberry production and consumption has been growing steadily in the last 20 years, and this is because it has been shown that consuming blueberries can help protect the organism from cancer, neurodegeneration, and obesity. My PhD project was on 4 main goals, and this is because this fast growth came with many challenges. However, today I'll just talk to you about the more agronomic side of my project, which was speeding up breeding and improving production. Speeding up breeding is very important because blueberry is a clonally propagated crop, meaning that going from pollination to a released cultivar can take up to 15 years. So having molecular markers and similar technologies available can significantly speed up this process. And then I mentioned efficient production. And this is because the cost of producing blueberries is extremely high, and 50 to 80% of the total production cost comes from handpicking those berries. Now, this is especially important in the US, where growers have to pay about 70 cents per harvested pound of blueberries using domestic labor. Now, domestic labor is scarce and thinning, so a lot of growers have to rely on imported labor. Now, we know with the current situation and administration, even this kind of labor is becoming hard to come by. And this is really determining a situation of crisis for growers, especially if you take into account that neighboring countries with similar growing seasons and that are really competing for the same market niche can pay their labor a fraction of this cost. So a solution that has been suggested as a way to restoring competitiveness in the US, like in many other countries, is using machine harvest, which would lower the cost to about $0.3 per pound. Now, machine harvest comes with a very firm requirement. That is, that the plants that are used by growers produce very firm blueberries that can withstand this very harsh physical handling. And so this is why I decided to focus on firmness for my project. As scientists, breeders, or molecular breeders, we have a responsibility to our stakeholders. So firmness really allows us to have a great impact on all of our stakeholders. It allows growers to have access to machine harvest. It allows suppliers to have access to berries that get less damage and so they spoil less. It allows consumers to have access to berries that are more enjoyable because consumers associate firmness with freshness. And so it really allows us to have a lot of impact throughout the whole supply chain. So just to give you an overview of how I decided to address this research question, I decided to go after the genes that are responsible for firmness. And to do that, I decided to genotype and phenotype a population, run a QTL analysis, and identify a few candidate genes. Now, these genes will be later used for some fine mapping efforts, and as soon as we have those final answer as of which ones are those genes that are responsible for firmness, then we can do some genome editing for breeding or for research purposes. So let me tell you a little bit more about the firmness phenotype itself. Firmness has been a major breeding focus for breeders for a long time. So what you see here are two graphs that exemplify exactly that. Each dot is a released cultivar. Red dots represent northern highbush blueberry cultivars. Blue dots represent southern highbush blueberry cultivars. The y-axis is the average firmness of a released cultivar, and the x-axis is the year a certain cultivar was released. So the two take-home messages from these figures that we elaborated in the last years is that the later cultivars tend to have better firmness than older cultivars, and this is because breeders have really been making a lot of effort in this trait. And also southern highbush blueberry, the subdivision of blueberry that we work with, has really been gaining a lot of firmness in the last years. And this is because we really focus on machine harvest as one of our priority traits. And after this, I started looking at the genetic architecture of firmness. And I started doing that looking at the gene expression patterns of genes that are involved in carbohydrate catabolism. And this is because firmness is really intertwined with cell wall integrity. Cell wall is also very intertwined with enzymes that degrade the cell wall. That is carbohydrate catabolism genes. So the ones that really attracted our interest throughout the ripening process are the ones that you can see at the top right of this picture. So for example, pectate lyase genes. So these are genes on which we designed guide RNAs for gene editing, and we will be verifying whether editing these genes affects firmness or not. Now, this is one of the projects that we're working on, but also we wanted to know the exact genomic location of other firmness genes. And so to do that, we decided to adopt a QTL analysis approach. So we used a population that is coming from a cross between two rather firm varieties. However, this population segregates for firmness, and this firmness variation needs to be captured in a QTL analysis. So we genotyped and phenotyped these 250 individuals, and we really phenotyped these 30,000 blueberries over the course of 2 years. But we also wanted to look at some other traits that are involved in machine harvesting. So we looked at firmness retention, and to do that, we measured the firmness of blueberries. We put them in a refrigerated environment to simulate shipping conditions. And then we took them out after a couple of weeks, and then we measured their firmness again. And also we looked at fruit detachment force. This is because when the machine goes in the fields and it shakes the bushes, we want ripe berries to fall in the trays underneath, and we want unripe berries to stay on the bushes to optimize our harvest. So we also want the differential fruit detachment force to be as large as possible, so we only get ripe berries into our harvest. And so for this, we only harvested and phenotyped 15,000 berries per year, and we only have results for 1 year, so these results are considered speculative. And so then one of my colleagues created a tetraploid map, which is quite an innovation in the field, using the markers that are interspersed in the genome that you see here. And this map is what we used to run the QTL analysis. So what you see here is the QTL analysis for the firmness traits. What made us really happy is that we could see a conserved peak for the firmness trait in 2018 and 2019, meaning that this is potentially a true peak. And also we could find one for the firmness retention trait. Then, as I mentioned, we also looked at the ripe fruit detachment force and delta fruit detachment force traits. We did find some peaks as well, but as I mentioned before, we only had 1 year of data, so these results are speculative. So then we went on and we looked at the candidate genes in these genomic intervals. For firmness and firmness retention, based on the literature that has been published on blueberry, we decided to focus on calcium and cell wall modifying enzymes. For fruit detachment force, we mostly based our work on what is published in tomato, because tomato is a berry and it's actually very well studied, unlike blueberry. And so for fruit detachment force, we decided to focus on METS-box transcription factors that have been implicated in fruit abscission, and also we have decided to focus on phytohormone signaling. So just to illustrate these processes a little, the reason why we decided to focus on calcium is because cell-to-cell adhesion is mediated also by molecules called pectins. Now, in their natural state, these molecules are usually highly hydrated and ramified. Now, when there is a higher concentration of calcium, pectin molecules create stronger crosslinks in between them, effectively creating a semi-rigid gel in between the cell membranes. And this results in increased firmness in the fruit. So we did find 4 genes that we thought were very interesting. 2 were related to calcium transport and 2 were related to calcium responses. Then, as I mentioned, we decided to take a look at genes involved in cell wall disassembly. So this is just a schematic representation in which the grid would represent cellulose fibers. Now, there are a lot more components in a cell wall, but cellulose is one of the main ones. And so there are some enzymes called cellulases which can degrade the cell wall. And so as a result, the cell wall gets looser and it will result in a less firm fruit because the structural integrity of the cell starts falling apart. So we did find quite a few of these genes and they have been reported. Now, they could have various functions, so this provides us a great basis for further studies in the future. For fruit detachment force, we decided to focus on transcription factors. Transcription factors, as you know, are proteins that migrate to the nucleus and activate or repress the expression of certain genes in response to stimuli, and then they activate cascading responses in the organism. In this case, it's the detachment of the fruit after the creation of the abscission zone in the pedicel-peduncle junction. As I mentioned, this is a well-studied process in tomato, and we found 2 genes in the intervals that we thought were very good candidates. The first one is fruitful, and the second one is agamous-like 15. The reason why we're so interested in these genes is because, in general, their overexpression blocks dehiscence or maintains juvenility, meaning that the fruit's not really detached. And also, fruitful was found to be similar to macrocalyx, which is also another gene very strongly implicated in fruit detachment. And lastly, as I mentioned, ethylene is very important in fruit detachment because ethylene is very important in the whole ripening process. So we decided to highlight 2 candidate genes that have also been studied in tomato and have been found to be important for firmness as well as fruit detachment. So just to wrap it up, we surveyed the firmness of most blueberry cultivars publicly available right now, and we investigated the gene expression of carbohydrate catabolism genes. And then we built a map for tetraploid blueberry and identified QTL peaks for at least 4 traits. And then we found and investigated numerous candidate genes for future investigations that we will be working on in the foreseeable future. So before I take any questions, I would like to thank the UF Plant Molecular and Cellular Biology Program and the FFAR Fellowship Program and the many collaborators that contributed to this project. And with that, I thank you for your attention and I'll take any questions that you might have. And to the bottom right, you can see my contact information in case you have any follow-up questions. Thanks.
So yeah, thank you, doctor. So do we have any questions? So yeah, I guess there are no questions right away. So yeah, as mentioned, if you have any queries, you can also contact Dr. Francisco. Yeah, thank you for your presentation. It was neat and clear.
Thank you so much. All right, just allow me a second to find out how to stop sharing my screen. All right, looks like I
Just
was
click
able.
on button.
All right,
Yeah,
thank you.
thanks a lot. Yeah, moving on to our next presentation, we have Dr. Narendra Kumar from Amity University, India. Dr. Narendra Kumar, are you available? Like we can see you online, so just wanted to confirm if you're available. Okay. I guess maybe Dr. Narendra Kumar is not online right now, but yeah, it's actually visible to us. So anyways, yeah, moving on. Can we have our next presenter, Dr. Camila Moura Santos from Maturium Startup, Brazil? Dr. Camila, are you available? Like, can we have your presentation?
Yes, I'm ready. Just a minute.
Sure.
Just a minute. I will
Definitely, sure.
share share the presentation.
Yeah. Your screen is perfectly visible to us.
Okay. Just a minute.
Sure.
I'm hearing a lot of feedback.
Oh. Actually, I'm sorry to interrupt. Like, we have a lot of noise or disturbance from your side. So could you mind just lowering any system beside you or something?
Yes, I just wait a minute. I will go to another place because of the
Okay,
noise.
no problem.
Ah, muito barulho, sei lá que porra. Hi, the noise stopped.
Yeah, the voice has stopped. Now it's clear. We
Okay,
can proceed
I'll
with the
initiate the
presentation.
presentation.
Yeah, sure.
Okay, thank you.
Hello. I'm really sorry to interrupt. Dr. Camila, actually your voice is not audible, and also if the presentation is not yet ready, we can move on with Dr. Narendra Kumar, and then we can come back to you. Is that okay?
Okay, it's okay.
Thank you. Thank you for
Okay,
your
ma'am.
information.
Dr. Narendra Kumar ready for presentation, ma'am.
Thank you, Dr. Narendra Kumar. Just a moment. Uh, Dr.
Ma'am,
Narendra
okay,
Kumar, you can
ma'am.
actually click on the screen button so that you'll be able to share
Okay,
your
ma'am.
screen with us.
Okay. It's not working.
Like, yeah, you
The
can just
screen
click on
button
the
not
screen.
working. Please allow me, ma'am.
Sure, sure. You can take your time. I'm sorry.
Okay, ma'am. Okay. Ma'am, is display visible on your side, ma'am?
No, it's actually not visible.
Is my display visible?
Not visible. Your display is not visible
Okay.
to us. Yeah, now it's visible.
I have just shared the screen.
Yeah, now it's visible to us.
Now it is visible. Okay, ma'am. Thank you. Thank you, Madam
Thank you. You can
Collins,
start your
for
presentation.
giving me chance to present my research work. Sorry, yeah. Okay, my topic of research is antifungal, insect repellent and fumigant activity of clovile for safe storage of rice seeds. Myself, Dr. Narendra Kumar from Amity Institute of Biotechnology, Amity University, Haryana, India. Okay, ma'am. Camera on me. No, it's not working. Loss of food commodities due to pest infestation is a major reason of food crisis. In spite of improvements in hygiene, it has been estimated that around 30% people in industrialized countries suffer from foodborne diseases each year. In 2000, 2 million people died worldwide. So, new methods are needed for reducing foodborne pathogens, possibly in combination with existing methods. So, definitely, we need new methods. Industrialized societies appear to move towards green wave consumption. They are desiring fewer synthetic food additives and products without harming the environment. That means major emphasis is reduce use of synthetic food additives, even reduce the use of synthetic products without harming the environment. The versatile composition of essential oils having broad spectrum of antimicrobial potential, showing low toxicity, have enhanced their widespread use in perfumery, pharmaceuticals, cosmetics, and even food preservatives and additives. They are capable of inhibiting foodborne pathogens and extend the shelf life of processed food. They prolong the storage life of food by preventing rancidity and oxidation of lipids. In this connection, if we deal rice is a major dietary component of people in most of the countries. This is consumed in Asia and Africa, but less in Europe. Rice is the most widely preferred cereal in the world. It is a staple food for two-thirds of the world population. The rice contains 80% carbohydrate, 7 to 8% protein, 3% fat, and 3% fiber. This rice staple food provides 700 calories per day per person for about 3,000 million people in the world. Rice seed is typically stored for 6 to 8 months after harvest, but more than 90% farmers don't take any precautions to protect it. Its proper storage enables farmers to earn high profit margins. Most farmers sell their rice seeds in village, local, and urban markets. Approximately 20 to 30% of stored rice food seeds become deteriorated by fungi and insects. The non-scientific storage of rice in rural areas here in India leads to heavy deterioration of rice seeds by fungi and insects. But detailed studies on such deterioration of stored rice food seeds have not been made so far. For their management, we have only alternative, that is pesticides. We are managing our rice food seeds with the help of pesticides. Here in India, I have observed, gone through various peoples are dying with the use of these pesticides. It is very, very toxic. They are using sulfas. So their management is done here in India with the use of chemical pesticides such as phosphine and methyl bromide. Insect resistance to phosphine is a global concern now, and control failures reported in field situations in countries. Methyl bromide being broad spectrum fumigant, they are declared as ozone depleting substance, and this is now phased out completely. In view of problems with current fumigants, there is a global interest in alternative strategies, developing of chemical substitutes, exploitation of controlled atmospheres, and integration of needs some physical methods. So definitely we have another alternative, that it should be through botanical management. That means first management was through with the help of pesticide, store seeds of rice with the help of pesticide, and second, store seeds of rice with botanicals, which is a plant organ. So definitely among botanical extracts used as a pesticide, essential oils are promising alternative to chemical pesticide. Nowadays, there has been interest in plant products, that is essential oils for fumigant action, since it is believed that natural compounds from plant sources may have the advantage over conventional fumigants in terms of low mammalian toxicity, rapid degradation, and local availability. That means these have these parameters. These parameters are very important because it's showing first parameter I told, this is low mammalian toxicity, rapid degradation, and local availability. This is locally available also. Plant is locally available also, and it has low mammalian toxicity. So the use of volatile antifungal substances for the control of disease caused during storage have been recognized to have a better future over other fungicides. So therefore, this study was undertaken for searching a renewable, biodegradable, non-phytotoxic, and harmless botanical pesticide to control the storage loss of food seed of rice. Therefore, mycological analysis, insect pest analysis were done. Samples of rice seeds were collected from grocery stores of Gurgaon, Gorakhpur City Market, India, and they were examined for associated fungi and insects. The technique adopted was agar plate of Muscat 1948, and this was the blotter method. 1963 and revised in 1970. Overall, mycological analysis revealed the presence of 15 fungal species. In the Blatter method of study, this was 13, and agar-plate method, 10 fungal species were observed. During the investigation, on the basis of percent occurrence, the most dominant fungi was Aspergillus flavus, Aspergillus niger, and Carbuleria lunata. Only one species of insect was observed, that was Cytophilus oryzae, popularly called here in India rice bevel. This is the image of dominant fungus, that is Aspergillus flavus. This is now Aspergillus niger, which is most dominant and effective. So the insect is rice bevel. Normally, I have observed it attacks more in the month of July and August in stores, even in farmer stores, even in grocery stores, or at any store level. This normally attacks more in July and August month. This damages the adult insect grub feed on the grain by making a small hole. Infested resource seam can be recognized by the white earths on the seed surface and the round exit holes with the help flap of seed coat. So the deterioration tests were done. Deterioration caused by dominant fungi, that is Aspergillus flavus, Niger, and Carbuleria lunata, which were dominant on the basis of percent occurrence, and the rice bevel with respect to weight loss and seed germination was done. Aspergillus flavus and Aspergillus niger played a very important role in seed weight loss and seed germination. Their protein content were studied. The protein content in Aspergillus flavus inoculated seed was 4%. Aspergillus niger, it was 4.5%. Carbuleria lunata, it showed only 4.6%, while the control set without fungus showed 8% protein content. Again, second parameter was studied, that is carbohydrate content. This is in Aspergillus flavus inoculated seed was 40%, Niger showed 43% occurrence, while Carbuleria lunata, it showed 44% carbohydrate content, and control set showed 75%. So it's a variation, and these fungal species are doing major role in deterioration of seeds, like in terms of its biochemical constituents, that is carbohydrate and protein content. These species are reduced. So I may select these organisms as a test organism, and this also showed definitely this reduced germination percentage also. So therefore, for developing an alternate, for developing an botanical, 100 plants were screened for their essential oils through Clevenger apparatus, and they were evaluated against all these three dominant fungal species, that is Aspergillus flavus, Aspergillus niger, and Carbuleria lunata. The techniques adopted by inverted pretty plate technique of Boker 1938. So out of 100 plants extracted essential oils separately tested, only one plant oil, that is Syzygium aromaticum oil, clove oil, shows complete inhibition. Its oil was characterized by its MIC minimum inhibitory concentration. The minimum inhibitory concentration of active oil was found to be 400 ppm. The oil was fungistatic at 400 ppm. This showed fungicidal nature at 500 ppm, and its spectrum of oil was also studied. It inhibited 412 fungi at 400 ppm, and at 500 ppm it inhibited all 15 fungi. This is the clove figure of clove diagram. It is so. Now, is effect of physical factors on oil activity were done. The temperature treatment up to 100 degrees centigrade was given. It showed no adverse effect on the toxicity of the oil. Autoclaving was also having no effect. Storage period of 120 days was given, no effect. Increased inoculum were also tried. 5 fungal disc of 5 millimeter and 1 fungal disc of 11 millimeter were tried. This showed no effect on the activity of iron. This was characterized with the physicochemical parameter. Physicochemical properties of clove oil were also studied. Showing the specific gravity 0.9559, specific titration +10, refractive index 1.540, acid value 3.45, saponification number 153.49, ester number 150.04. Phenolic content in this oil was found to be nil. This was its solubility were also studied. This was completely mixed with petroleum ether, acetone, and 90% ethanol in 1:1 ratio, but this was found to be insoluble in water. This was found to be highly insoluble in water. In due course, some advanced study like GCMS analysis were done, revealed the oil revealed 16 compounds. The major compound was usinol, that is 55.60%, usinyl acetate 20.54%. This was followed by caryophyllene 14.84% and alpha humulene. This was 2.75%. This is the oil, clove oil, and this is the aluminum phosphide and ethylene dibromide. These are the some their diagrammatic presentation how the aluminum phosphide looks, and this is the figure. So definitely some thing about the aluminum phosphide. It is well known. This can affect if you breathe in. If in contact, irritate the skin and eye, nose, throat, and lungs, causing coughing, wheezing, and shortness of breath. Reported exposure may damage the lungs, kidney, and liver. Aluminum phosphide reacts with water, moisture, and releases highly toxic and flammable oxygen gas. Ethylene dibromide is liquid at ambient temperatures. This can again cause irritation to skin, eye, mucous membrane, and upon breathing of the lungs, liver, kidney. These effects can result from any routes of exposure. The systematic effects of ethylene dibromide are in part due to metabolic conversion in the cell, toxic and 2-bromoacetaldehyde. Persons with pre-existing skin disorder, eye problems or impaired liver, kidney or respiratory tract infection may be more susceptible to its effects. So therefore, in vivo investigations were done. Now we have for storage of rice food seeds, we have two alternate. One using of synthetic fumigants, ethylene, aluminum phosphide and ethylene dibromide, and second is botanical, that is clove oil obtained from Syzygium aromaticum. Containers were tried were tin containers and the concentration maintained was 1000 ppm and 1500 ppm, and they were kept for storage up to 3 months. But I strongly say that oil treated samples show no fungus. This showed complete protection. Aluminum phosphide treated samples show 6,4, and in agar-plate method, blotter may it showed 6 and 3 fungal species respectively. While ethylene dibromide treated samples showed 8, 7, and blotter method again showed 8 and 7 fungal species respectively. Control set was having 8 fungal species. Through both the techniques adopted, that was agar plate and blotter. After 6 months of storage, again this was container used was tin and the storage period kept 6 months and the concentration was same at 1000 and 1500 ppm. Aluminum phosphide treated samples showed 13 and 11 fungal species, while Blatter had 7 and 5 fungal species. Ethylene bromide again in agar technique 13-13 respectively. Blatter 13-13. And the control set are having 16 fungal species. But here again the Cesium aromatic amyl completely control the rice food seeds. That means they are going to provide complete protection. So phytotoxic studies were done. Phytotoxic effects after 3 and 6 months of storage were done in tin containers. When stored in tin containers, after 3 and 6 months of storage, there was no effect on protein and carbohydrate content. There was no effect on seed germination, seedling growth, plant growth, foliage morphology, general health of plants. Seeds storage were done in gunny bags. Again, aluminum phosphide treated and ethyl dibromide, both fumigants, synthetic fumigants were used. They were stored in a container like gunny bag, and the storage period was 3 months. All treated sample again so no fungal species, that means it's provided complete protection. In agar plate technique, 7-4 species, and through blatter it is 8 and 6 fungal species respectively. Ethylene dibromide treated sample source through agar plate technique 8 and 7 fungal species, and blatter it is 8 and 8 fungal species. Control set was having 13 fungal species. After 6 months of storage, concentration again kept 1000 and 1500 ppm. Global completely protected. That means no fungus. It showed complete protection. Rice seed, there was no fungal species after this much of storage time at this concentration. Aluminum phosphide treated samples shows 10 and 7, Blatter 8 and 7 respectively. Ethylene bromide in agar plate 10 species, and Blatter here after 6 months of storage in gunny bag, ethylene dibromide was found to be completely ineffective. Control set show 13 fungal species. Technique adopted were agar plate and Blatter technique. So after phytotoxic studies were done after 3 and 6 months of storage in gunny bags. There was no effect on seed protein and carbohydrate content, percent germination, seedling growth, foliage, general health, and morphology of the plant. So now the conclusion session. The essential oil of clove can be exploited for preservation of rice food seeds during storage. First thing is strong fungicidal nature, broad range of activity, capacity to withstand heavy inoculum density, strong repellent activity against rice weevil, persistence of activity at high temperature, long storage period, doesn't affect seed protein and carbohydrate content, doesn't affect seedling growth, doesn't affect general health and morphology of the plant. So thanks to Emma Collins ma'am for giving me a chance to present my work. Thank you very much, ma'am. If any question, ma'am, please.
Thanks a lot for your presentation, Dr. Narendra Kumar. It was informative. And do we have any questions? Do we have any questions? So I guess there are no questions. Anyways, thank you for your presentation. And yeah,
Okay,
moving
ma'am,
on to
I
our.
need a photograph, ma'am. Till you're not on the set, I need a photograph, ma'am.
Oh, actually, we use desktops and we do not have any webcam, so that's the reason we couldn't on our cameras. I'm sorry for that.
Okay, okay.
Yeah, like usually we only conduct regular conferences for which we do not need any webcam. So yeah, that's the issue over here. We're sorry for the
Okay,
inconvenience. Yeah.
okay. Thank you very much for giving me a chance to present my work, research work.
You're welcome. Thank you for your interest in participation too. So
Okay, thank you very much, ma'am.
thank you. So yeah, moving on to our next presentation. So Dr. Camila, are you actually ready for the presentation?
Yes, I'm ready.
Oh yeah, thank you. Just a moment, I'm just making you the presenter. Yeah,
As are you seeing the
yeah,
presentation?
yeah.
Okay. I'm gonna present to you an improved protocol for efficient transformation and regeneration of Cetar italica. This is part of my work in my PhD that I finished last year. Are you hearing me?
Your voice is
Hello?
actually very low. It's not that clear.
Okay, I'm gonna try
Yeah,
speak loud.
yeah,
Um,
sure.
Well, Cetar italica is a cereal group well adapted to grow under stressed environments. It's important for healthiness and food security. And this species is an interesting alternative for small scale grain production in a global change scenario. Wait a minute, it's not changing the. No, okay. It has some interesting characteristics like relative small genome. It has a short life cycle, a short stature, and numerous seeds production. It's an interesting alternative for studies with C4 metabolism. Well, this species is considered by scientific community a recalcitrant species because it's hard to develop protocols for its regeneration from callus. There are some articles with good alternatives, but they reach until 10% of efficiency in the transformation. The DNA delivery technique is a key of the transformation process. For monocotyledons, it's well used the transformation by agrobacterium, but there are some problems with the regeneration systems because the protocol of regeneration medium needs to be really specific for the species to make that this regeneration occurs. So our aim was develop a protocol of transformation of Cetar italica with the intent to assist the effort of Cetaria scientific community to develop a method of agrobacterium transformation based on early development based on mature seeds. We developed this protocol based on earlier developed by Cetaria viridis. So we used the protocol of Cetaria viridis as a base for develop the protocol for Cetaria italica. For this work, we used two vectors, one containing a GFP gene sequence and another an RNA interference sequence to test the transformations. We used the Agrobacterium strain EHA 105 and the cultivar of Cetaria italica UG1. For the confirmation of the transformation, we made segregation analysis. For the events transformed with GFP, we used fluorescence microscopy. And for the events that we used the RNA interference vector, we made analysis of gene expression. This is the pipeline of the method. It is divided into 6 main phases. That is the phase of co-induction, the phase of inoculation and co-culture, the phase of selection, shoot regeneration, elongation and rooting, and also acclimatization. The first step is the callus induction step. We use it, as I said, mature seeds that we sterilize it and we inoculate into a callus induction medium that I'm gonna call as SIM for 5 weeks in the dark at 28 Celsius degrees. Then we isolate those callus and we subculture those embryogenic callus into a fresh medium, SIM, for 1 week more, again in the dark at 28 Celsius degrees. And after this last week, the calli were prepared to the transformation. The infection step lasts around 4 days. We prepare agrobacterium solution and it reaches between 0.9 to 1.0 of OG for the transformation. We also prepare the calli with a treatment of ice bath. We use this treatment to reduce the defense response of calli and its oxidation. This bath takes around 20 minutes. We use the co-inoculation medium to make this bath before the step of infection. The co-inoculation time we do around 30 minutes at 26 Celsius degrees. And then the next step is the calli drying. We make this process to reduce the contamination by exacerbate growth of agrobacterium. We put those callis in a petri dish with a stereo filter paper, and it dry at 15 minutes, more or less 15-20 minutes. Before and after that we co-cultivate those calli on another plate with co-inoculation medium, also for 3 days in the dark at 22 Celsius degrees. Then we pass to the selection step. In this step, the infected cali are transferred to callus induction medium containing timentin. It stays in this plate at 1 week in the dark again at 28 Celsius degrees. After this week, we change the medium, the plate. Now the medium has timentin and hygromycin. The Kali stayed in this plate for more 1 week in the same conditions. Well, after that, we went in the regeneration step. It takes around 4 weeks. In this, we first tried the medium of regeneration of Setaria viridis for Setaria italica, but it doesn't work. You can see this in the pictures. The first picture is show a callus of Setaria italica that stayed around 1 month in the medium regeneration of Setaria viridis, and we have no differentiation. The picture in the side shows the callus of Setaria italica in this new regeneration medium that we developed for the species. The main changes that we made in the medium for Setaria viridis is we changed the synthetic cytokinin. In Setaria viridis, we used the kinetin, and then we change for 6BA. After the change of the phytohormones, we start to see some differentiation, but it was very clear that the plantlet wasn't developing well, because it seems that it was suffering by drought stress. Then we reduced the sucrose content to 1.5% and accented glucose to ratio 1:1. In addition, we put hydrolyzed casein to help the development, the regeneration. And then with this change in the protocol, we reach a good rate of regeneration of those calis. Another change that we made is that we introduced activated charcoal pills inside this plate with the aim to capture the ethylene produced by the callus and reduction the moisture inside the plate, avoiding the precipitation of the steam on the walls of the plate and avoiding contamination. Before the regeneration phase, we went to the elongation and root stepping. This phase, we put the regenerated kali in a pot of glass with root medium for the species. Now you see those pictures. You can see the development of the callus. Here is in the beginning at around 2 weeks in this medium until the 4th of 5 weeks. It's when the plantlet is already formed. Then we pass to the phase of acclimatization. This phase, we put the plantlets in a thawed period period of 16 hours light and 18 hours dark, with intensity luminous of 600 micromoles, again at 28 Celsius degrees, and around 50% of relative humidity. Yes, this is the regeneration efficiency table. Here we test the regeneration of Setaria italica callus in the medium developed by Setaria viridis and also in the medium that we developed for Setaria italica. Wait a minute, it's yes. The first two lines show the results of tests done with 170 cali placed on plates with the regeneration medium for Setaria viridis. Note that there are no regenerated plants from those cali. And the two bottom lines shows the results of the test done with 213 Kali placed on plates with the regeneration medium that we developed for Setaria italica. Okay, now this is the transformation efficiency table. This transformation protocol for Setaria italica reached 40% of efficiency with an average of 19.2%. It was an increase of more than 100% of efficiency when compared to other protocols available. Samples of genomic DNA from regenerated plants were tested for vector sequence integration by PCR analysis. Here you can see in the figure the bands of the expected fragments referring to the hybromycin resistance gene. The confirmation was present at 41 events of 43 plants tested. One of the bottlenecks of our protocol transformation is the relation of the number of Kali per volume of agrobacterium suspension. In this table, we can note that lots with the highest number of Kali resulted in the lowest percentage of efficiency in some of the transformation process. So this indicated us that the relation of bacterial units per Kali has a great influence on transformation efficiency. Here we can note the localization of GFP fluorescence in the leaf blades and roots of transgenic plants transformed with GFP vector. The first picture shows the transverse section of leaf blade that belongs to a non-transformed plant. Here, as I don't know if you can see. And the transverse section of a leaf blade that belongs to transformed plants. And this picture here shows the root segment of a non-transformed and of a transformed plant. Well, the analysis of gene expression was performed with the second generation plants that were transformed with the RNA interference vector. Here we can see the reduced expression of the target gene in transgenic plants. And for this experiment, the expression level of the target gene was normalized by 2 housekeeping genes by FOL method. These are the results of segregation analysis. About 60 seeds of transformed plants were segregated with selection medium to obtain the first generation of the transgenic plants. And segregation analysis was performed with using the chi-square test. The table illustrates the chi-square results. All events presented a segregation ratio of 3 to 1. So these results confirms the integration of the transgene into the plant genome, as well as the stability. So, as a conclusion, we can affirm that the new protocol developed showed a high rate of efficiency in DNA delivery and also in regeneration of fertile transformants. The transformation was stable over the generations, and this protocol can provide an interesting tool for Setaria community research in relation to transgenic work with Setaria italica. Well, I would like to thank you all for the attention. Thank you for the opportunity to participate of the Congress Plant Genomics. Here below is my email if you want take some doubts. I'm glad to respond your questions. That's it. Thank you very much.
Hello? Yeah, I'm sorry. Am I audible?
Uh,
I'm sorry, it was on
Yes,
mute.
okay.
Yeah, I'm sorry, I was just talking and it was on mute. So thank you for your presentation. Do we have any questions?
You're welcome.
So I guess there are no questions. Anyways, thank you for your presentation, Dr. Camila, and also for your time. So we hope to meet you further.
Thank you. Thank you very much.
Yeah. So I just want to confirm, is Dr. Srinivas Janaswamy from South Dakota State University, USA is available online? Because I could see some 2 to 3 logins without any names. So I just want to confirm if I'm not missing out. So no, I guess Dr. Srinivas is not available. So moving on to our poster presentations now, we have Dr. Phan Thi Hong Nong from Catholic University of Louvain, Belgium.
Yes, I'm available. Thank you.
Thank you. Just a moment. I'm just making you the presenter so that you will be able to present.
Uh,
Yeah, you can actually click on the screen button so that you'll be able to
uh,
share your screen with us.
yes,
Yeah,
can
that's
you see
perfect.
it?
Yeah, it's perfect.
Okay, just a moment because I want to
Sure.
collapse. Sorry. Sorry, just a moment. Wait for me. Just a moment. I will make a
Yeah,
full
sure.
screen.
Sure, no problem.
Sorry, I tried to share file in full screen, but I maybe I lost in somewhere. So just wait for me 1 minute, I think.
Sure. In the go-to meetings, you can just click on the screen so that your sharing will stop. And once again, click on screen so that you'll be able to choose whichever option you want to show us.
Okay, just a moment.
Mm-hmm.
Oh, yeah. Excuse me, can you move on to another person and I will come back later?
Okay, okay, no problem.
I
Yeah,
think
we
I
can
need
do
to
that.
restart the app so I can come back later.
Sure. You can just
Yeah.
click on the screen button so your screen sharing will stop.
Uh,
Like in the go to meetings button, you can just click on screen. So anyways, in the meantime, Dr. Sonu Bharati from National Institute of Technology, India. So Dr. Sonu Bharati, are you available? Dr. Bharati, are you available right now?
Yes, ma'am, I am available.
Yeah, please click on the screen button so that you'll be able to share your screen with us.
Okay. Hello.
Oh, yeah. You
Good
can
evening
actually
to
click on the slide share button. Like if you're not feeling comfortable, you can just click on slide share and you can just zoom in and out so that it'll be easy for you.
ma'am. Actually, I'm facing some problem displaying.
Uh, like beside the volume button, you have a view like slideshow. You can just click on that.
Ma'am, actually, I have a single poster only.
Yeah, that's perfect. That's all right. Like it'll show you full screen and you can just zoom in so that it'll be clearly visible to everyone. So that's what my idea was. Or you can simply save this as a PDF if possible from your side. So you can just zoom in and out for your convenience.
Um, Ma'am, is it visible now?
Yeah, it is visible. Perfect.
Uh, Ma'am, actually, my work was based on terminally Arjuna, population structure, linkage disequilibrium, and marker trait association for arginolic acid in terminally Arjuna. So here I am showing the biochemical part. So my poster is based on agroclimatic zone based identification of elite terminally Arjuna accession concerning to arginolic acid production. So in this graph, I'm showing the scenario of global herbal market in terms of geography. So Japan contribute $9.8 billion, that is 16% of total economy, and Asia 10.8, and rest Europe 2.5, 2.4, European Union 28 billion dollar, that is 45%, and others 4.1. And my objective to identify the accession of terminally Arjuna with high amount of arginolic acid content, represent the different agroclimatic regions of India, and quantification of arginolic acid by HPTLC method. So for this study, I collected 140 accession of terminally Arjuna represent the different agroclimatic zone of India. So this is the map of India, and these are the states from where I collected my samples. That is Uttar Pradesh, Jharkhand, Uttarakhand, Assam, Chhattisgarh, Odisha, Andhra Pradesh, and Maharashtra. And these a total of 140 accession were selected and obtained for bark patch for extraction of arginolic acid. For the extraction of arginolic acid, first we made 200 milligram of bark powder exactly, and then transfer this bark powder in a conical flask, 100 ml conical flask, and mix with 20 ml of ethyl acetate and kept for 10 minutes for pre-leaching. Then this conical flask was irradiated in microwave at 65 degrees for 5 minutes. That is in intermittent way. 1 minute is cooling, 1 minute is irradiation, then 1 minute for cooling. This is for 5 minutes, and then filter out this suspension with the help of Wattman filter paper number 1. And the emulsion were cooled with the filtrate, and the solvent was vacuum evaporated at 40 degrees centigrade. The solvent-free residue was dissolved in 1 ml of HPLC grade methanol and transferred to a 1.5 ml Eppendorf. Then HPLC processing and estimation of arginolic acid in the bark extract. Pre-coated aluminum coating silica gel plate purchased from Sigma Ridge and used for this study. So for this purpose, HPTLC system component and software, that is Chemic TLC scanner 3 instrument, was used. And HPLC silica gel plate of dimension 20 centimeter into 10 centimeter was used. A 20 ml of freshly prepared mobile phase. The composition for 100 ml of mobile phase is 60 ml toluene, 30 ml ethyl acetate, and 1 ml formic acid, and methanol 10 ml, and 200 ml derivatization solvent present, that is composition for 100 ml, 85 ml ice cold methanol, 10 ml acetic acid, and 5 ml sulfuric acid, and 1 ml N-acetaldehyde were used. The derivatized sample fingerprint was scanned at 595 nanometer on Chemic scanner. So these are the fingerprint. Derivatized sample fingerprint at 595 nanometer. So on the agroclimatic zone based, and this is the arginolic acid percentage. So I collected 140 samples. These samples represent 5 agroclimatic zone of India and 9 states. So on the basis of agroclimatic zone, so there are 5 agroclimatic zone. These are Central Plateau Hill region. The maximum content of arginolic acid estimated in 1.443% in Central Plateau and Hill region, that is maximum, and minimum in Western Himalayan region, that is 0.995, and on the basis of state, hello,
Hello?
hello, ma'am, am I audible?
Yeah, you're audible. Sorry, it was on mute.
Ma'am, on the basis of state, the Assam has minimum content of minimum percentage. The population collected from Assam had the minimum content of arginolic acid, that is 0.06%, and maximum from the population collected from Uttarakhand, that is 0.203%. And
I'm really sorry to interrupt, Dr. Sonu Bharti, but your voice is actually not audible. Just wanted to confirm.
Okay, ma'am, something network problem. Ma'am, this is the phytochromatogram of HPLC plate of arginolic acid standard, illustrated by TLC scanner. We use different concentration of standard, but we use for this result is 100 microgram per ml standard of arginolic acid. So 8 microliter standard working stock was used, and this is the photochromatograph, and on x-axis represent the retention factor of each detected spot, and y-axis the height of the peak, and z-axis location on the plate. So this is the HPTLC graph of arginolic standard illustrated by TLC, and this is the photochromatogram of the HPTLC plate of Arjuna back extract of sample 45 illustrated by TLC scanner. So response factor was estimated between 30 to 40 seconds for the arginolic acid. And this is the histogram showing the percentage of arginolic acid on the basis of agroclimatic zone, and this graph showing on the basis of state. So maximum content of arginolic acid was estimated in the population collected from Uttarakhand and minimum from the population collected from Assam. And on the basis of agroclimatic zone, so maximum content of arginolic acid estimated from the population collected from Western Himalayan region and minimum from the Eastern Himalayan region. So key finding of this is of this research is the on the basis of agroclimatic zone, Western Himalayan region had the significant highest for arginolic acid. In contrast, Eastern Himalayan region recorded the significant lowest value for arginolic acid. And on the basis of state, accession from the Uttarakhand state exhibit significantly the highest value for arginolic acid. On the other hand, the accession from Assam state had significantly the lowest value for arginolic acid. And on the basis of 140 accession collected from the different locations and from different agroclimatic zones, the accession UKDT-LP from the Uttarakhand and Western Himalayan agroclimatic region produced the highest amount of arginolic acid, and the accession APMD03 from the Andhra Pradesh of Southern Plateau and Hill agroclimatic region, the lowest amount of arginolic acid was estimated in this accession. So on the basis of this result, we can selected the superior examplars for the maximum content of arginolic acid, because arginolic acid is very high in pharmaceutical industries, because most of the drugs for cardiovascular disorder made up by the arginolic acid or triterpenoids, saponins. So we can multiply these accession with the high arginolic acid content for multiplication of the exemplars with the high content. So thank you.
Thank you for your presentation, Dr. Sonu Bharti. So do we have any questions? Do we have any questions? Yeah, if you're having any questions, you can please ask. I guess there are no questions. Thank you for your presentation.
Thank you, ma'am.
Yeah, and also for your time. So Yeah, moving on to our next presentation. So, Dr. I'm sorry, Fan Thi Hong,
Yes, I'm
from,
ready.
yeah, one moment, please.
Yes.
Yeah, please click on the screen button to share your screen with us.
Okay. Can you see it?
Yeah, that's visible.
Thank you very much to give me a chance to present about my research. Today I would like to present about my research is identification of quantitative trace locus controlling soil tolerant trace in rice at seedling stage by GWAS. First of all is introduction. A lot of research focused to study about the soil mechanisms in rice and as well as the effect of sun on rice. So we can see on several pictures in the screen, picture A, B, C, and D, the symptoms of rice plant under salinity conditions. So we can see that salt cause the dehydrate uh, in youngest leaf, and they cause the disorder in the old leaf, and then we can see a lot of plant died. And in the paddy field, in the paddy saline soil, I took the pictures 10 years ago, so we can see this several plants can survive and a lot of plants died. So the main purpose now we need to improve the finding the new varieties resisted saline condition, and how to improve is the big questions, and as where the genetic information is lacking. And we know that the the gene Sontaran is controlled by multiple genes, and we know several important genes. We know that salt causes the osmotic effect and then ionic effect and disorder. The osmotic effect is the symptom is quite similar to drought or the other stress. However, the ionic effect is very unique for salt stress. And the other research about the genes. So we detect that more than 100 genes related to salt tolerant, and almost genes detected by BLN linkage mapping populations. So we compare the two methods how to detect some QTL or genes. In the left side is the parental linkage by crossing some parents, and then we cell population or by cross and so on. So, and finally we analyze and find some QTLs in the progenies. So this method provides the high statistical power but very low resolution because limited the limited combination. However, the new methods is genome-wide association study, in short we call GWAS, with a lower statistical power but very high resolution because it's natural population with a long history of crossing, cell pollination, and so on. So it's very diversity and it's good to detect the new genes. And now I move on to the second part is the material method. In my research, I analyzed the GWAS with 2,391 assessments, and it includes in indica group, japonica groups, ost group, aromatic group, and some admix. Admix here we is crossed by with indica and japonica, and we don't know it belong japonica and indica. And I only I saw the genotypes in the different hosts, and then after 3 weeks in hydroponics, I evaluate the sun stress by SES. SES is the standard evaluation system, a standard evaluation score with the score 1, 3, 5, 7, and 9. Meanwhile, number 1 is the most tolerant and number 9 is the most sensitive. And in the pictures is about the place which I conducted my experiment. In the left side is the picture I took in the sowing time. So in different hosts with a different genotype, each host with each genotype. And for one plate here, I cultivated with 720 hosts with 720. And the experiment will be was laid out by augmented design. And in the right side, we see the two boxes. With one in the left side is the control with no salinity, and in the right side is with salinity is 60 millimole of sodium chloride. So we can compare and see the different in phenotype. And then I analyze GWAS with by the software I call fast LMM factors petrolate transformer linear mix model, and I download the genotype data from 3K project. And after that, I remove the mineral iron frequency more than less than 5% and missing rate less than 5%. And after that, I call almost 600,000 SNP. And after, because the capacity of the computer, so I only random the 40% of genotypic data to analyze, like genotypic data for analysis. The other ones is I random 1% to calculate genetic similarities, and I calculate the principal component analysis, and I choose three first component analysis, three first component to as the covariate data, and then combine with the phenotyping data, and we find some QTLs, and then we find some significant SNP, and then based on the LDDK, we conclude some QTL. The next main part is about the result of my experiment. So in the figure 1 is the PCA plot of the first PCA, PC1 and PC2 of 2,391 assessments and some groups: APMIS, aromatic, OS, indica, and japonica. So in the figure 1 in the left side, we can see the different corners like a triangle. So different corners with the purple color is japonica, the blue color is indica, and the with the green colors is also is the three angles with the difference. So it means that when we analyze GWAS, the population structure is very necessary to consider. And in the right side, the figure 2 is about the LDDK of genotype data of whole population and different subgroups. So from here, we determine some LDDK of different groups. Meanwhile, indica is the faster, then OS, then whole population, and then japonica, and the biggest is the aromatic. Figure 3 is about the frequency distribution of a standard average score in sun tolerant of different groups. And from here we can see because we have a lot of individuals in indica, right? So in the figure the figure of frequency, we know that indica have a lot of individuals with more than 1,422 assessments. And during in the phenotypic data, we know that the different subpopulations have a very similar. I mean, in general, we have means value is not too different. And then we collect some, we analyze and plot with the Manhattan plot and QQ plot. And if we see the QQ plot, we can see that the model I chose in this research is quite fit. So maybe as data is good for the model and it's quite fit, so we can use this model for our result. And in the left side is about Manhattan plot of whole plant with the 2,391 genotypes. Then with indica panels with 1,422 genotypes, with Japonica panel with 650 genotypes, osmotic os panel is 182 genotypes, and with aromatic panels with only 65 genotypes. And after I set the thresholds about the loss score is about 4. So we detected some QTLs, and I mark with the red and green color. Meanwhile, green color and black color. Meanwhile, black color is the novel QTLs, and the green color is some report. It contains some reported genes related to Sontaran was previously reported. And from here, we based on the result, I make a summary about some detected QTLs, and we detect some QTLs with 4 QTLs in chromosome 1, 2 QTL in chromosome 3, 1 QTL in chromosome 4, 5 QTL in chromosome 6, 1 QTL in chromosome 8, 1 QTL in chromosome 10, 1 in chromosome 11, and 3 in chromosome 12. And in this, among the QTLs, we detect 6 QTL contain in interval contain some previous QTL or genes related to Sontaran. For example, chromosome 1 contains some located in the long chromosome in a long QTL, conferring the sodium chloride, sodium concentration, or potassium concentration, ratio of and sodium and potassium in leaves, in plants. And the other genes in chromosome 6 contain some genes inside related to Sontaran. For example, OC-SAPS8 is a member of stress-associated protein, and here controlling Sontaran. OS8AK10 is the high affinity potassium transporter in rice, and is the main result. And after that, I make some small conclusions. Based on our result, I conclude that the LDD case, based on the LDD case, we know that the LDD case of indicas is the fastest, and then followed by osmotic, the anti-population, japonica, and aromatic. Based on the result of PCA, we know that it will show the genetic diversity, and we can divide in the 3 main clusters, like indica group, japonica group, and os group. And based on the result of GWAS, we know that the models were well fitted to the data, and 18 QTLs were detected. Meanwhile, we have 6 QTL were located in interval containing 2 genes controlling Sontaran, and 12 the other QTLs is novel. And from here, we know the perspective that we will analyze the haplotype, analyze this, and then we find some candidate genes that the future work I should do. And about my result, and in this research, I would like to thank to the RSCC Day to support the financial support for us for our research, and thank you to the members in our lab in Earth and Life Institute, Agronomy in University Catholic de Leuven in Belgium, and also thanks to my college in Vietnam National University of Agriculture to help me to finish the research as well as to analyze GWAS. And in the future, I hope we can find some interesting candidate genes related to Santorin and hope give some informative data to the breeding in the future. Thank you very much.
Thanks a lot for your presentation. So do we have any questions?
Oh, hi, I'm Monica. I want to do a question.
Sure, you can go on.
What percentage of the traits are explained by the QTILs?
Excuse me, I cannot. I cannot hear. Well, can you repeat?
What percentage or quantity of the traits are explained by the QTIL that you discover?
The score?
I don't know if my English is not so good.
No.
Sorry.
I think it's my fault. Yeah, I detected some QTL and I set a loss score about the number 4 is mean 0.0001. Is the p-value? Did you ask about the p-value, right?
I asked you about how proportion of the salt tolerant phenotype
Okay, yeah.
is explained by the QTIL.
Yeah, okay. Sorry, I think it's here. The salinity level, right? I said about the 60 millimole of sodium chloride and is equivalent about the 0.3%.
Okay.
Yes, about.
Thank you.
Yes, thank you.
Thank you.
If you have anything or any questions, you can send me by email and I will reply.
Thank you very much.
Yes, thank you. Thank you so much.
So do we have any other questions? I guess there are no questions. Thank you, Dr. Fan Thi Hong. It was really informative, and also thank you for your time.
Yes, thank you.
Moving on to our next presentation, we actually have presentation of Dr. Panumas Kotipong from the Department of Agriculture, Thailand. So yeah, Dr. Panumas is actually not available, so I'll be just displaying his presentation on his behalf, and you can just look at it. And if you're having any queries, I'm sorry I'll not be able to answer your questions, but yeah, you can send an email directly to them and they'll be able to answer your questions. So yeah, this is a presentation. And yeah, actually you can move, you can zoom in and out so that you can read it clearly. So I'll just present this for next 3 to 5 minutes. So you can just take a look. And if you're having any questions, yeah, you can just send them an email and we'll be providing you their email IDs. I'll not disturb you any further. Yeah.
Hello.
Hello?
Ma'am, this presentation is half is disappear. So how I can see the full PowerPoint, but this poster.
Oh, I'm sorry, I didn't get you. Can you please repeat it again?
Okay. Ma'am, I am asking that ki, full poster is not displaying. Half is displaying. So how I can see full poster?
Okay, okay, I'm sorry for that. So could you please provide me your email ID in the chat box so that I'll send you an email? You can just check that out.
Okay. Okay, sure.
So once this like after 2 to 3 minutes, so once the session is completed, I'll send it to your email if you don't mind.
Okay. Thank you.
Thank you. Thank you. So if everyone is has done reading, I can just change. We can just move on to the next presentation. So yeah, moving on to our next presentation, we have Dr. Muhammad Al Farouk on behalf of Dr. Srinivas Janaswamy from South Dakota State University, USA. So yeah, are we ready for the presentation, Dr. Muhammad?
Yes, I am. It's Buster, by the way.
Oh yeah. Yeah, okay. Yeah. Just a minute.
Okay.
Yeah, you'll be able to share your screen. Just click on the screen button. I'm sorry for interruption. We do have just to inform everyone, you can just use the zoom in and zoom out buttons if you're facing any difficulty in the visibility. So yeah, you can zoom up to 400% over here, and you can just move up and down the screen so you'll be able to see clearly whatever the information you would like to see. Thank you.
So should I explain anything or wait for questions?
Yeah, you can explain. You can explain your part, and then we can wait for the questions.
Okay, so at the beginning, I'm not a doctor. I am a PhD student. Dr. Srinivas Janswamy is my supervisor, and this is a part of my PhD work. So we are trying to study the effect of cyclodextrins and sodium chloride salt in the gelation of iota-carrageenan. In this study, we used two different concentrations of sodium chloride with the three types of cyclodextrins, which is alpha and beta and gamma cyclodextrins. And before I explain more in this regard, I'd like to explain what carrageenans. So carrageenans are a group of polysaccharides that extracted from seaweeds, and mainly there are about 15 types of carrageenans, and the three famous ones are lambda, kappa, and iota carrageenans. In this research, we choose to use carrageenans, and also we used another type of carbohydrates, which is cyclodextrins. In this case, we would like to study the gelation properties of the carrageenans because we would use it later as coating materials to encapsulate bioactive component. So it's important to know the gelation properties because also we will use it as material to prepare fibers, and those fibers we will use them later to encapsulate bioactive component. To be able to prepare those fibers, we need to study the physical properties of the gel and the thermo stability of the gel. So for that, we aimed to study the rheological properties and the thermal stability and microstructure, and also studying the water mobility and some other physical properties. So in the physical or in the physicoelastic properties, we study the temperature effect on the gel's behavior and the frequency sweep at different temperatures. And the table number 1 is presenting the physicoelastic properties and gelling temperature of those mixtures, then followed by studying the water mobility of the gels. Then we studied also, as it mentioned in table 2, the thermal stability to make sure what will happen during the process of using this gelling to encapsulate bioactive component. Also, we studied the microstructure to make sure we will be able to make those fibers. So I will be happy to answer any questions if the audience have any questions.